So today's topic is amino acids. Okay. First things first, what is an amino acid? Normal definition is it is a building block for proteins that constitute the body. Okay. This is a good answer, but along with it, you lend it two additional lines that it has an amide group, an alpha carbon and a carboxylic group. Then it makes the entire sense. So there are various kinds of amino acids. They play various different roles, and there are many things that we will go through in the lecture. Okay. After this lecture, you should be able to tell yourself what an amino acid is, what are its kinds, what are its types, what are its functions, and how you can differentiate between them. This question often appears, or a part of it often appears in your university examination. Especially, which one? The one that they might ask you classification of amino acids. Okay. There are definitely short notes on the CONH2 bond or the peptide linkage, and definitely they are going to ask you what are essential amino acids, non-essential amino acids, semi-essential amino acids. Okay. They will ask you these questions if they do not ask you the gross big classification for amino acids. Clear? Okay. So this usually amino acids have this structure. There is a carbon C, and this is called alpha carbon. Very good. This is called alpha carbon. There is a COH group, there is an NH2 group, and there is definitely an R. There is definitely an R, and this R represents any group. This R represents any group that can be present in the amino acid, giving you a very diverse array of functionality to choose from when you are forming proteins in your body. Now, where have this amino acid come from? Dropped in the rains? Blew in with the wind? Where did it come from? This is a very easy question. You have all given NEET examination. It comes from living animals only. From where in the living animals? Each and every cell is producing amino acids. What kind of amino acids? That can be produced in your body. The other kind of amino acids you are absorbing from nature via diet, via food. Okay, you are taking it via food. So this is where the amino acids are coming from. That is the source. Now, how these amino acids is being produced in a living cell? Remember anything called central dogma? Can you kindly tell me what a central dogma is? Yes, very good. So there is a DNA that gives you RNA, and that RNA is translated into proteins. And during this process of translation, the amino acids get stuck one with another with a peptide linkage to give you a complete protein. Which is thereby getting used in your body. So, after this lecture, you are supposed to know standard and non-standard amino acids, essential and non-essential ones, glucogenic and ketogenic ones, and how the side chains, this R is going to play an important role in defining the character of that particular amino acids. Okay? Very good. Now see, proteins are made up of hundreds or more. This is how the chain looks like. After the amino acid class is done, we will get into the concept of protein. You see, this is a linear structure. So linear structures are how many Ds? 1D. 1D, right? A straight line is a 1D. What is a straight line? The minimal distance between two points. Okay. So this is 1D structure. Do amino acids, do proteins look like that? No. How that happens we will teach you, but we will teach you later. Like now we will address the elephant in the room or the amino acid in the room. Chalo. So genetic code, you have heard of genetic codon, triplet codon. More than one amino acid can be coded by more than one triplet codon. Okay, and that is how it is being generated in the human body. This is a central dogma. These are the genes, transcriptions, mRNA is formed, mRNA is being translated, and it will form proteins with specific amino acid sequences. Clear till now? Any doubts? Anywhere you got lost in the road of life, chasing a black cat or something, or addressing the elephant in the room perhaps? If you lose track, you do tell me. Okay. So general structure, very simple. Alpha carbon. What you mean by alpha carbon is that the carbon has 4 different groups attached to it. It has got 4 different groups attached to it. That is alpha carbon. There is a COH group, a carboxylic group, an amino group, and this R is different for each and every different amino acids, giving it special structure and nature. Okay. Standard amino acids and non-standard amino acids. There are 20 amino acids that are universally coded. Okay. 20 amino acids that are universally coded, whether that is essential or non-essential, it does not matter. Okay. But it will be coded by your central dogma. It will be coded by your what? Amino acid being coded from where? RNAs. Okay. It will be coded from your RNAs. You know these things. What you unlikely are to know, there are some non-standard amino acids. Okay. So what are these non-standard amino acids? They basically appear fleeting. They come for a moment. They are initial substrate of a reaction. They appear and go. They appear, they form their work, and they go. Okay. So these are intermediates of the metabolic reactions that are going into your body. You will not find them freely existing in nature. You will not find them freely present in nature. And there are some special type of amino acids. The first one is being selenocysteine. Selenocysteine is a human being made amino acid. Okay. It's a human being made amino acid where instead of the stop codon, we are using SACIS, selenocysteine insertion sequence. We are using or calibrating your translation process to put a selenocysteine there, that is a selenium containing cysteine, where the sulfur is being replaced by selenium in the sequence of your amino acid. And then there is pyrrolysine. This is usually found by methanogenic archaea or bacteria that is found in volcanic hot water domains. Okay, volcanic hot water domains. They help it to produce methane. They help it to produce methane. Okay. So standard amino acids, alpha structure. This is the alpha structure. This is the alpha carbon. This is the R group. This is the COH group. This is the amino group. Okay. So these are called standard amino acids. All of you know D and R, that has been taught, D and R. So normally that R configuration, okay, the R configuration is usually present in nature. The R configuration is usually present in nature. Now pay attention here. They have a primary amino group and a carboxylic group substituent on the same carbon atom. They are present in the same carbon atom with exception to proline. Normally, the COH group and the amino group are independent like here, but proline looks like this. So, its amino acid has got two bonds, first with the supposed alpha carbon and second is along with the R group to form a cycle. That is why it is called an amino acid. It is a variation from the standard structure. Okay, that is why it is called amino acids. Now this amino acid gives proline a very unique property. It is very tensile. If you stretch it, it will stretch. When you leave it, it will regain its conformation. Just like a spring. What this helps is in protein structures, in loops and bends, which are basically the supporting structures, they give role in support just like columns of a building. Just like columns of a building. They have comprensile strength. Okay, they have tensile strength due to their unique structure of the nitrogen. Okay, due to the unique structure of the nitrogen. Now non-standard amino acids, first is selenosis, that has already been discussed. Other classifications of amino acid, non-protein amino acids, okay, ornithine and citrulline. These are usually found in the pathway while producing urea. Okay, they appear fleeting during the metabolism. They come, they get used up to form something else. Just during the reaction they are present, they are not found naturally in the nature. Okay. Then there is non-alpha. That means the NH2 group is not attached to the alpha carbon. The NH2 group is not attached to the alpha carbon. How that happens, I will teach when I am teaching the specific functional group. What I want you to concentrate on this, modified amino acids. Okay. So modified amino acids usually are proline. Okay. They are proline and hydroxylysine. Proline or hydroxylysine. They are usually present in your joints. They are usually present in your joints and they undergo a reaction. They become hydrated. They become hydrated. They become hydroxylated to form hydroxyproline and hydroxylysine, and this imbibes water. You had taught mucopolysaccharides in the last class, right? So there you know it can imbibe water and it can let it go. It can imbibe water and it can let it go. Exactly the same thing is happening with hydroxyproline and hydroxylysine, thereby giving you tensile strength in your joint and cartilages. Okay. Now, this is the short note that you are gonna get. If you are lucky enough to not to get it in your university examination theory, you will definitely have it in your practical. Essential, semi-essential and non-essential. So, essential is you directly take it via food. Okay. You directly take it via food. You cannot synthesize it in your body. You do not have the enzymes. You do not have the capability. Then comes non-essential amino acids. They can be synthesized from the amino acid that you have got from diet. That means your body has the specific enzymatic capacity to convert one amino acid to another, so that you do not need to absorb more than 8 essential amino acids to live. Rest, you have the capacity to generate in your body. You basically transfer one amino acid into another kind. Here, now this is the funny part. This will always be asked and you will always give the correct answer each and every time in your life when it will be asked. You know why? Because I have got your back. A. Hill. Oh, let me rub it, otherwise I will not have enough space. A Hill MPTs TV. A Hill MPTs TV, a master of physiotherapy who is residing in Hills, he has a TV. A Hills MPT TV. Okay. So for A, you have got arginine. For A, you have got arginine. If I find someone saying alanine in your viva, I am going to strangle you. A for arginine, not alanine. Alanine is produced in your body. A for arginine, not alanine. Now H is histidine, I is isoleucine, L is leucine, L is lysine, M is, any ideas? Methionine. Methionine. P is phenylalanine. T for threonine. T for tryptophan. And finally V for valine. And that's it. That's all, folks. Looney Tunes. A Hill MPTs TV. Remember this mnemonic forever in your life and you will never make a mistake out of it ever. Be it your UG vivas, be it your PG vivas, be it your PDT vivas. A Hill MPTs TV. That is how easy this is. A Hill MPTs TV. And rest, alanine, aspargine, aspartate, cysteine, glutamine, all of these can be produced in the human body. Okay. This is another classification on the basis of metabolism. This is another classification on the basis of metabolism. What it means is some amino acid can be broken down into glucose. They can be broken down and you can form glucose in your body via a metabolic pathway called neoglucogenesis. Neoglucogenesis. So these amino acids that have the capacity to give you glucose, they are called glucogenic amino acids. And some of them give you keto groups, that is acetyl CoA or acetoacetyl CoA. It is acetyl CoA or acetoacetyl CoA. What is acetyl CoA? We will delve into it later. But they give, these are the ketogenic identities in the human body. They are the ketogenic identities on the human body. Right now, mug it up. Right now, mug it up. It will make sense later. Okay. So, some amino acids give you keto entities like acetyl CoA or acetoacetyl CoA. Some of them give glucogenic amino acids. They can be converted into glucose to fuel your metabolism. And the rest are mixed. They give both. Now, there is a very easy way to remember this also. Very easy way to remember this also. Ketogenic amino acids, remember lysine and leucine. Remember lysine and leucine. LL. Keto L. Okay. Ketogenic are lysine and leucine. Glucogenic are arginine and histidine, both being basic amino acids. Both being basic amino acids. Arginine and histidine. And rest at rest. So, in order to answer this question, you need only remember one thing. LL, lysine and leucine are your ketogenic, arginine and histidine, both basic amino acids are your glucogenic, and the rest give both. out of all 20 amino acids. Out of all 20 amino acids. This is the final stay of your viva when things have gone bad. This is the final stage of questioning in your viva when things have got bad. So you absolutely cannot afford to bumble here. Very easy. A Hill MPTs TV, LL, lysine, leucine and arginine, histidine. That's all. That is going to put you back in the safety net. That is going to pull you out inside the safety net. So these two are the most cardinal and important questions that will arrive in your university exams and your university vivas and whatever exams that you are further going to give. All will have this part. No one can deny it. And you will always be able to put it down easily. A Hill MPTs TV, LL and arginine, histidine. Okay. Now, amino acids can be classified in various ways. There are various ways to classify it. And all of these classifications, some are very important, some are somewhat important, but it is good to know. But it is good to know. Now, this classification is generally being done on the basis of the R group. The R group which is changeable for each and every amino acids. Okay. On the basis of the functional group, they can be classified as aliphatic, aromatic, acidic, basic, acidamide, sulfur containing and amino acid. Example of amino acid? Proline. Very good. Example of amino acid is proline. And based on the side attachment, they are classified as alpha, beta, gamma. Good to know. Just hear it and try to keep it. Please don't click photos. You will get the entire thing. When you click photos now, it gives me a glare. I get distracted. Whatever you only require to hear me out, and then after the class, take the PowerPoint presentation. If you click photos, if you pull up your mobile, that distracts me very badly. Okay? That distracts me very badly. So please don't do it. Please don't do it for any faculty. They will give it to you. This has been prepared to be given to you. I have been through this long back in 2008. It's all to give it back to you. Okay? So please do not pull up your mobiles. Please do not click your pictures. You have a recording going on. You can go and watch it anytime in your college website. Okay? Chalo. Now, amino acid classification is also based on hydrophobicity or hydrophilicity. Now, you have to tell me what is hydrophobicity and hydrophilicity. What is hydrophobic nature and what is hydrophilic nature? Tell me. Yes. So, water, all people will not be one. Some people are good friends with someone. Some people are good friends with someone else. Whenever you put two together, you will find that huddle. Okay? So, things where you are huddling one side is hydrophilicity. You have the same nature. And the nature is you like water like hippos. You like water like hippos and bisons. The another sector does not like water. They will go away from water. They are the cats of the group. Okay? They are the catty people of the group. So, hydrophobicity is if you put it in water, it is soluble and it will form a homogeneous mixture. Hydrophobic is it will stay away from water, it will try to shell itself and push the water away from its central zone. That's all it means. And all of them have various natures. All of them have various natures. Now, there are hydrophobic amino acid which are non-polar and hydrophilic amino acid which are polar. Non-polar means they are aligned in such a way, though they are electrically neutral, they have a tiny bit of magnetic alignment. There is a tiny bit of magnetic alignment. They cause a magnetic field due to the spatial orientation of the different functional groups that are present. They are electrically neutral. If you test the electric neutrality, you will find it 0. But due to the presence, the distance between them, distance of the groups, they will exert a magnetic field. This is known as your polar. Non-polar is electrical neutrality 0 and magnetic neutrality is also 0. Okay. So that is what non-polar and polar means. Now non-hydrophobic amino acids that are non-polar are usually the alkyl group containing amino acids. The alkyl group containing amino acids where the R of the amino acid is an alkyl group, like CH2, like CH2CH3, like hydrogen, like isopropyl group, it has a R group. Okay, so that is alkyl group. And then there are aromatic ones. Aromatic ones, they form a ring. They form a ring. You will not find a benzene ring here. Come on, ask. Proline aromatic में proline aromatic में नहीं डाला because proline has a heterocyclic group. It is a heterocyclic group. When I am saying aromatic group, it means a homocyclic group. Okay, proline is a heterocyclic group because the N has 2 bonds, one with the carbon of an alkyl group and one with the alpha carbon. That is why proline has not been placed in your aromatic group. Okay, and then there are hydrophilic amino acid polar group. They can be neutral, they can be acidic, they can be basic. The statement only tells you why it is polar. Okay, the statement only tells you why it is polar. Basic acidic amino acid means it has got 2 COO group. So if it has got 2 COO group, then it will definitely have a different electrical effect. It will have a different electrical effect. Same with basic group. There are two NH2 groups. And neutral is tyrosine, serine, threonine, cysteine, asparagine and glutamine. Okay. Now let us see how they look like. First comes glycine. It is the simplest amino acid and it is a university long question. Glycine, write about its structure, write about its bonds and write about its metabolic interactions. That will be covered in amino acid metabolism. For now, no, this is a long question. For now, no, this is a long question. Glycine. So here is the entire arc part and the fixed part, more or less fixed. That is your alpha carbon, the carboxylic group and the NH2 group. So if you put an H, it becomes a glycine. If you put a CH2, it becomes alanine. If you put 2 CH3 groups on 2 sides of the carbon alternate to the primary to the alpha carbon, then it will give you leucine. It is much more easier to understand if I draw it. Alpha carbon, COOH, NH, very good. H, give you glycine. CH2 gives you alanine. Now put a CH3 over here and a CH3 over here. Gives you, it is as easy as that. Now put a CH2 over here and put a CH3 over here, then you will get isoleucine. You will get isoleucine. So hydrogen, methyl group, isopropyl group and branch chain isobutyl group on the gamma carbon. Do not delve into it. Okay, do not delve into it. So this is the alpha carbon, this is the beta carbon, this is the gamma carbon. Okay, right from alpha carbon is where your functional groups are attached. So alpha carbon, beta carbon, gamma carbon, and it will go on and forth till infinity. So do not delve in it. Just see that these three are your simple alkyl amino acids. These two are your branched amino acids. They are branched amino acids, and all of them are having alkyl group as their R. Okay. Now here, with side chains containing hydroxylic group, they have an OH group. The first is serine, second is threonine, and the third is tyrosine. Serine, threonine and tyrosine are your hydroxyl group containing amino acids. They are hydroxyl group containing amino acids. Now in serine, it is located just on the, if this is alpha carbon, this is the beta carbon. So the beta carbon, you put very simple structure. Put an OH over here, it becomes what? Serine. Put another CH2 and then a hydroxyl group over here, it becomes threonine. Tyrosine we will talk about later. Tyrosine we will talk about later. It is so easy. Just a change in the position and the placement of the OH group. Then there are side chain containing sulfur atoms. These are sulfur containing amino acids, and man, they are metabolically important. They are metabolically very, very important. Must come university question. Single carbon metabolism will appear in your university papers. No one can stop it. And the answer is done by sulfur containing amino acids. Okay. So what happens is, here this is OH. You remove the O and put a thiol group. SH is thiol group? Yes or no? Am I right or am I wrong? Boss is always right. Okay? Boss is always right. Even when he is wrong, he is still right. So, you just put a sulfur group in the place of oxygen, you put a thiol group in place of a hydroxylic group, and voila, you have got cysteine and methionine. You have got cysteine and methionine, which are your sulfur containing amino acids. Which are your sulfur containing amino acids. Now, these are the acidic amino acids. Acidic amino acids. Why acidic amino acids? They have got two carboxylic groups. They have got two carboxylic groups. First is aspartic acid and the second is glutamic acid. First is aspartic acid and the second is glutamic acid. Very good. Can anyone remember something? Glutamic acid, valine, glutamic acid, valine. Yes, sickle cell anemia. Very good. Sickle cell anemia. This is how you corroborate. What happens is, in the beta chain of hemoglobin, in the 6th position, in case of sickle cell anemia, this glutamic acid is replaced by valine. Now you know why you need to know this nature. Due to the presence of the COH, it is a charge structure that will give you a polarity. When that is being replaced by valine, which is a non-polar structure, during the acceptance of oxygen, which is a strenuous process, what happens is the RBC sickles because the hemoglobin clumps together. The hemoglobin clumps together. That is the problem. And why it clumps together? Now today you have come to know, because glutamic acid has two carboxylic groups, which gives it polarity. This is why biochemistry is important. This is how you are supposed to make sense of what you are taught. This is how you are supposed to comprehend the importance. Otherwise, you will feel lost. Otherwise, you will always think, I didn't sign up for this, but that is not the case. Sometimes I will be able to explain, sometimes I will not be able to explain, but just hold on through the journey and you will be fine. Now comes the basic amino acids. Now comes the basic amino acids. Arginine, histidine and lysine. Arginine, histidine and lysine. What are the special method about arginine and histidine? They were, yes, they were glucogenic or ketogenic or both? They were glucogenic. They were glucogenic. Okay, you can make glucose out of them during time of need, during time of starvation. During time of starvation, you can pick up glucose from there. Okay, so that is the good part about your arginine, histidine and lysine. These two are your basic amino acids. Now let us talk about histidine a little. Okay, let us talk about histidine a little. It contains a group called imidazolium group. Okay, it contains a group which is called imidazolium group. Now how this nice thing happens looks something like that. Alpha carbon, COOH, NH2, primary structure. Okay, now there is a large big group for arginine. I cannot remember it, sorry. I cannot remember it. Just remember there is a big group with another NH2 group attached to it. Now lysine, COOH, NH2. 1, 2, 3, 4. Alpha, then beta, then gamma, then epsilon. Then epsilon. Here you have got an NH2 group. Alpha, beta, gamma, epsilon. In the epsilon carbon, you have an NH3 group. Okay? In case of lysine, this gives it structural polarity. This gives it structural polarity. And finally come to histidine. It has a imidazolium group. It has an imidazolium group. Just put a CH3, put a pentagon with a nitrogen over here and your job is done. Okay. This is the basic structure, put a CH2 and then a pentagon with a N downside. This is your imidazolium group, which is the part of histidine, Please remember, in undergraduate levels, we do not expect that you will able to remember or draw structures. We do not expect, it cannot be done, it is an impossible job. I am drawing the structures in order to make an imprint on your minds that this should look like this. Okay? So if you are unable to remember the structures, if you are unable to draw the structures, know that you are not wrong, know that you are not fumbling up, it's just nature taking its course. Okay? It is not mandatory, neither it is essential. We need you to know the concept. We do not expect that you will be able to draw structures. So if you cannot do that, please do not fret, please do not get stressed. It is absolutely fine. So after histidine, we have got aromatic group or aromatic amino acids. Aromatic amino acids. The first being phenylalanine. Now if you put a CH3 group on top of a benzene, it becomes what? One answer, Loud and clear. 1. Toluene. Very good. Toluene. So if you put a toluene with this basic structure, you will get phenylalanine. If you put a toluene with that basic structure, it is phenylalanine. This is your basic structure. Put a toluene over here. Voila, you have got phenylalanine. Now if you put a hydroxyl group at its tail, removing this CH3, that can be done in organic chemistry, and I suppose you know how to do it. This becomes tyrosine. Is tyrosine an essential amino acid or a non-essential amino acid? Tyrosine is essential. Non-essential amino acids. A Hill MPTs TV, T for threonine and 3 for tryptophan, not tyrosine. Okay? Tryptophan, not tyrosine. This is, see, these are the two most common mistakes you are going to do. You are going to recite alanine instead of arginine, and you are going to spell tyrosine in case of, you will speak tyrosine in place of tryptophan. This is the only two honest mistakes that you are going to do during your universities. So be mindful of it. Be mindful of it. Okay. So this, and your tryptophan is a heterocyclic group. Your tryptophan is a heterocyclic group. So you put a methyl benzene, it becomes phenylalanine. You put a alpha hydroxylic methyl group, it becomes, or a phenol group, it becomes tyrosine. You put a heterocyclic group, it becomes tryptophan. It is so easy. It is so easy. COH, NH2, this is your base group. You put a methyl. After putting a methyl, you put a pentagon with a nitrogen that is below, and then you complete the cycle. And this is how it becomes tryptophan. It has got two heterocyclic structures. It has got two heterocyclic structure, and this is called indole group. This is called indole group. It will be very important during your amino acid metabolism. For just now, just be familiar with the terminology that your tryptophan has a indole group, and proline, as you already know, put an inverted pentagon, put an N, put an H, put a COH group on its side, and here you have proline, with this being the amino group. With this being the amino group. This is the part we have all been waiting for. What is the use of today's lecture? This is the use of today's lecture. We are finally there. The moment of truth. The moment of truth, why we have been toiling so hard. What are its uses? Okay, the final round. So first is aromatic amino acids. Aromatic amino acids. Phenylalanine. How does phenylalanine look like? How does phenylalanine look like, my friends? Tell me. You put a methyl benzene, it becomes phenylalanine. You put a phenol group, remove the CH3, put a OH on its tail, it becomes tyrosine. You put two cycles with uneven pairs, and they will give you the heterocyclic amino acid called tryptophan. It will give you the heterocyclic amino acid called tryptophan. Now phenylalanine. I told you why tyrosine is not essential. It can be produced from phenylalanine. Look, it is so easy. How I will tell you. First step of amino acid metabolism. Hydroxylation reaction. Remove the CH3, put an OH. And this enzyme is called phenylalanine hydroxylase. This enzyme is phenylalanine hydroxylase. And in deficiency of the phenylalanine hydroxylase, you have a genetic condition called phenylketonuria. That is why you have been working so hard, being patient for last 30 minutes, not throwing chalks and dusters at me and being quiet for this moment of truth. Okay? Now is it making sense? There you have phenylketonuria. It is a genetic disorder. It is an autosomal recessive disorder. It's an autosomal recessive disorder. Now similar to phenylalanine, it gives you so many neurotransmitters. It is the basic form of so many neurotransmitters that it is not even funny. And some of them are dopamine, maintains your normal blood pressure. Norepinephrine maintains renal perfusion of your kidneys. Epinephrine and adrenaline, which gives you flight, fright and fight metabolism during danger. So if your diet does not contain phenylalanine, what are you going to do? Okay, you can't do anything. You cannot produce these neurotransmitters. That is why they are so important. That is why they are so important. Now come to tryptophan. Tryptophan has a very nice reaction with the vitamin called niacin. Okay, that will be taught later. Now tryptophan gives you serotonin that calms your mind, that allows you to take serotonin. Then comes the hormone melatonin, maintains your internal biological clock, allows you to differentiate between day and night, makes you drowsy during day and long lectures and unhappy during night due to sleeping through the day. Okay? That is not how it's supposed to be. Come on, be a heart, have a heart, man. Okay? So, then alanine. It is the basic storage form of glucose which could not be stored in your liver like glycogen. The rest of it will be put into alanine and that alanine will go into your liver. So when you have finished with your glycogen storage, that is about 12 hours after fasting, then you need energy. You are going to pull that alanine out. The alteration of alanine cycle is linked to formation of type 2 diabetes, something we will discuss at the later stage. Okay. Then valine, a very important thing, a very important protein that does lots of work in your bone marrow. And valine is also the person which is a part of sickle cell anemia, Now comes leucine. What was the funny part of leucine? It is a branch chain amino acid. It is a branch chain amino acid. And if you are not able to metabolize these, then you have branch chain amino acid dehydrogenase deficiency. That will give you maple syrup urine disease. You will have ketoacidosis, you will have syrupy urine, you will have multiple stones and all that good things. You have all that good things which are the bread and butter for a successful urologist. Okay. So, leucine plays the most important role over here. Then there is methionine. One carbon metabolism precursor. It makes tyrosine from tryptophan. Thereby allowing you to produce 5-HT, 5-hydroxytryptamine, which is a neurotransmitter, which acts as a neurotransmitter. Threonine I don't like so much, so you can leave it. Then is histidine. Histidine and arginine. What are the special characteristics? They are basic. Okay, they are basic. They have got 2 NH2 group and they play the most important role in the buffering action of your blood. Okay. In most bad diseases or bad conditions, acidosis develop in the body. Okay, be it sepsis, be it respiratory failure, you will either have a metabolic acidosis or respiratory acidosis. Alkalosis do occur, but acidosis is more common in life-threatening conditions. So there, this arginine and histidine will allow you to maintain your blood pH. It will allow you to maintain your blood pH. Without maintaining your blood pH, you are likely to fall short of oxygen supply. All your partial pressures of oxygen and carbon dioxide pressures will become unsettled. It will become unsettled and she is so happy and sleepy. Good morning. Okay, pay attention. Okay, your blood pH is going low. You need arginine and histidine to charge you up. Okay. You need arginine and histidine to charge you up. Come on, I have also slept in my professor's classes. There is no shame in it. Okay, there is no shame in it. So definitely these are a little bit difficult, a little bit tipsy-turvy. No matter how much color I put on it, it is a very boring topic. Okay, at the end of the day, it's a very boring topic. So it's very normal for you to feel sleepy, but don't worry, I'm there to wake you up. Okay, so histidine and arginine, basic amino acids, and they maintain your blood pH by countering the excessive acidic condition when it appears in your blood. Okay, and with this we have talked about amino acids. With this we have talked about the chemistry of amino acids. Now I have got to talk about a much more important topic after amino acids, which is a must to know, and that topic is called isoelectric pH. It is called isoelectric pH. What is this isoelectric pH? Don't worry, it will be explained. Now imagine there is a basin. Now imagine there is a basin, okay? And in that basin there is a nice little sponge. Nice fluffy sponge. So many holes over here, so anything can go through it. Plenty of holes, anything can go through it, just like cottage cheese. Okay. Now in this portion you have made a pH gradient. What do you mean by pH gradient? It is right from -7 to +7, and you were able to do it because it is a sponge. At this part there is a different pH, at this part there is a different pH, at this part there is a different pH, and it is gradually increasing. It is gradually increasing. Okay. Now think, this poor fellow, this bechara fellow is a basic amino acid called histidine, the poor histidine. You dropped him over here. Bechara histidine, you have dropped him over here. He was sleeping beautifully into the night. You pulled him up, you put him in a plane with a paratrooper gang and dropped him in the sea. Okay. Now he has got 2 NH2 group. He has got 2 NH2 group. What will he do? There is a pH gradient. There is an NH2. It has got 2 NH2 group. It has fallen in foreign territory. It is in acidic pH. What will it do? Most likely nothing. It will sit over there. Most likely nothing. It will sit over there. Why? He is too fat and heavy for his own good. He cannot move. Now we have sent a rescue team. You have sent a rescue team. You understood that he is not the person where you are supposed to skydrop. And that rescue team is an electrical field. That rescue team is an electrical field. Now since he is in acidic group, what he will become, he will be polarized. He will be polarized. Now he has excess charge. He is a basic person in an acidic medium. And you have applied an electrical field on top of it. Now you are helping him to move. You have given him a support party to pull him out of the enemy lanes. So once this electric field is given, this fellow is going to move towards your where? Positive end, because here it is negative, here it is positive. It will move towards the positive end, and when he reaches the positive end where its electric charge has been equalized, he will no more move. He will sit down over there, and this is called isoelectric pH. This is called isoelectric pH or PI. It is the point where a protein or amino acid will stop moving in a solution when it reaches electrical neutrality in a pH gradient. Okay? Now, one amino acid can have one isoelectric pH, two isoelectric pH or three isoelectric pH. Okay? Because the highest number of groups you have is 2. Since this was a gradient, there were different pHs. So him being basic, if you have dropped him over here, you would have moved over there. Since you have dropped him here, you have moved over there. Okay, so this is the concept of your isoelectric pH, and one amino acid can have one isoelectric pH or many depending upon its R group. With this, we are done with today's lecture. Thank you for a very patient hearing.